CareDx introduces AlloSeq TX17, the next generation in genetic matching. It is an innovative NGS HLA typing solution that uses hybrid capture technology, which eliminates the inefficiencies of using traditional long-range PCR methods. AlloSeq TX17 goes beyond the traditional transplant-related loci to consider more transplant-associated genes and helps you to identify the best genetic match. AlloSeq TX17's improved laboratory workflow provides multiple different advantages. Let's look at each of these steps in detail. The first step of library generation is a 4-in-1 process, wherein for each sample well, genomic DNA is bound, fragmented, normalized, and tagged by the bead-bound transposons. This tagmentation is followed by the addition of index adapters. This process allows for less than 50 nanograms of input DNA. It then undergoes PCR amplification to create libraries that have unique indices and the whole genome data. This early indexing step leads to reduced risk of sample switching compared to long-range PCR methods and does not require any manual quantification of DNA prior to pooling. You then pool all of the sample libraries into a single tube for a single tube workflow, and also have the ability to add additional genes without impacting this workflow. The next step is hybrid capture. Let's understand how our regions of interest are enriched in this process. First, we add a biotinylated probe panel to our pooled library. The biotinylated probe binds to the region of interest and is then enriched using streptavidin magnetic beads. The regions of interest are then eluted to produce an enriched targeted library that are then ready for sequencing on Illumina platforms. After 17 to 24 hours of sequencing, our proprietary AlloSeq Assign software helps to evaluate the results. The uniqueness of AlloSeq Assign lies in the fact that you don't need any special hardware or software requirements to run it. It will show you the reads as they are without any modification. And most important of all, it has a really fast data import speed. With the right settings, you may be able to get the import speed of 1 minute per sample, saving you valuable time. With its innovative hybrid capture technology, AlloSeq TX17 offers advantages that long-range PCR does not, like the expandable gene content without affecting the lab workflow, and easy single-tube workflow with early indexing step. AlloSeq TX17 also offers improved allele balance compared to long-range PCR techniques, wherein one allele is usually favored over the other. Another advantage with AlloSeq TX17 is reduced allele dropouts. In long-range PCR, based on the primer design, allele may or may not amplify, resulting in the dropout. However, with AlloSeq TX17, probes bind and capture regions of interest, including bases that did not bind PCR primers, resulting in no allele dropout, thus confirming that AlloSeq TX17 will provide better performance through improved allele imbalance, reduced dropout rates, and no PCR artifacts or crossovers as compared to long-range PCR techniques. And finally, the fast import speed of up to 1 minute per sample and enhanced user interface that will save you time and effort proves AlloSeq TX17 is truly the next generation in genetic matching.